Hi everyone! Our group today will explain about microscope. First, let us introduce our members' name. Our group leader is Nurul Suhaila binti Muhammad Razi, and our gorgeous members is Nur Atasha binti Nur Azmi and Nadia Afifah binti Murad. And lastly, our cute members Nur Mulyani Atika binti Zahari and Nur Shazwani binti Cik Hashim. Our one and only man in our members group is Muhammad Amir Shauki bin Abdul Hamid. Okay, let's begin with the introduction of microscopy. Microscopy is the technical field of using microscope to view objects and areas of objects that cannot be seen with naked eye. There are a few types of microscope.
Okay, first let me explain about simple microscope. Simple microscope, also known as magnifying glass, it consists of single convex lens. It is used to produce an enlargement image of the sample.
Next is compound microscope. It is used to view smaller specimens such as cell structures. Compound microscope consists of structural and optical components. It has two convex lenses, the objective lens and the eyepiece. Next is confocal microscope. Confocal microscope have superior resolution, much higher than conventional optical microscope. It is primarily used in measurement application. It uses a laser as a light source. Confocal microscope enable the non-contact, non-destructive measurement of three-dimensional shape.
Do you ever heard of scanning electron microscope? Scanning electron microscope produce image by scanning the surface with a focus beam of electron. It produces 1 until 20 nanometer resolution. The entire column need to be under vacuum, and the special part of scanning electron microscope, it form black and white image. Okay, this is an example of slide under scanning electron microscope.
Next is transmission electron microscope, TEM. Microscopy technique in which a beam of electron is transmitted through an ultra-thin specimen, interacting with the specimen as it passes through it. It will produce flat 2D image. It also produces high resolution. It is required use of an interface such as fluorescent screen, photographic plate, or electronic display.
Next, we will go through with fluorescent microscope. Fluorescent microscope is an optical microscope that uses fluorescent and phosphorescent. It is to study properties of organic and inorganic substances. The specimen is illuminated with light of a specific wavelength. Lastly, stereo microscope. Stereo microscope is known as dissecting microscope. It produces a three-dimensional image. It is used to study the surface of solid specimens. It is widely used in manufacture. Instrument uses two separate optical parts with two objectives and eyepiece.
Now, let's continue with the body parts of the microscope. First is the eyepiece. Eyepiece is function for what you will look through at the top of the microscope. The objective lenses, the primary optical lenses on a microscope. Arm. The function of the arm is supports the tube and connect it to the base of the microscope. Stage. The stage function as the flat platform to place the slide. The focus control. It is function to focus the specimen and move the stage up and down. The stage clips. It keeps the slide from moving around on the stage.
Automatic.